Welcome, everyone. We're going to get started right away. My name is Baron Kutcher. I'm on the Board of Governors of the Alumni Association, which has been a really great thing to do. I can encourage you to think about it. It's an amazing way to meet some really, really bright, energetic, interesting people. We're honored to have Dr. Jean Tang here. Dr. Tang actually is true Cardinal, graduated medical school in '99, PhD in '03. Stop me if I'm wrong on any of this. Completed her residency in '07 and is now a professor of dermatology. Her area of expertise, which she'll be describing, but I just think it's extremely interesting. The big picture is that she works on personalized gene therapy to treat rare diseases. So we're going to be learning a lot from her. She's published in the New England Journal, JAMA, and Lancet Oncology. I can say I particularly appreciate what she's doing. I'm an ophthalmologist, and we see really devastating ocular surface problems, especially, and blinding problems that are related to the types of diseases that Dr. Tang is exploring. So, and we really need help in finding better treatments. It's continues to be a very, very difficult problem to treat. So, please join me in welcoming and thanking Dr. and Professor Jean Tang for being here.
It's so nice to see you guys. I hope you can hear me okay. It's a very small, informal room. As I was coming here today and walking through some of these beautiful new buildings, I certainly didn't take classes in these buildings, and I think most of you did not, right? A bunch of us were in trailers or in the room 103, 104 hallway buildings, right, with the dean's courtyard. So as I walk through these areas, I think, boy, these students have it good. And apparently on the second or third floor, they have a gym just for the medical students to work out. And so look how far we've come, but I hope coming back to campus brings you a lot of good memories. I walk by certain areas and I think of when I took exams or step one, and I still get, nervous chills from that experience, and I think you must as well too. So, even though many of us are physicians or family members of physicians, there are some skin wound images that are a little bit intense, and so I apologize for that. But I'm glad I'm not speaking during lunch. So what I really wanted to tell you about, and the theme of this meeting, is how do we move technologies and discoveries from Stanford out into industry and commercialization. And we heard about these, incredible AI agents and incredible drug discovery approaches. I wanted to talk about gene therapy and rare skin disease. I study a rare disease called epidermolysis bullosa. Many of you may have never heard of it. It's rare. It's 1 in 100,000. And basically, the individuals lack a protein called collagen 7. Collagen 7 binds the epidermis to the dermis. When the baby is born and taken out of the mother's womb, you could already see blisters. So any sheer pressure or friction lends to blistering and open wounds. And these collagen 7 molecules make these staples called anchoring fibrils. So what do we do for these kids? They're known as butterfly kids because the skin is as thin and fragile as butterfly wings. And so they have blisters and wounds, and sometimes the wounds heal, but over time the wounds become quite big and they don't ever heal. And the babies and kids are wrapped head to toe in gauze and bandages just to try to protect that skin layer. So we use bandages, we use ointments. Because of a lot of bleeding, they have anemia, they have cardiovascular disease, and they have corneal erosions leading to scarring, GI problems as well, because the epithelium of the GI system is affected. And so what we wanted to do is to heal the wounds, and we knew that there was a single mutation. All we needed to do is put back the gene called collagen 7. And we'll tell you a couple of approaches on how we did it. And I went into medicine always being excited about genetic medicine. Now there's gene therapy drugs for sickle cell, and I just thought this is the lowest hanging fruit. If there's a single mutation and we can correct the mutation or put back the protein, we got to be able to fix the disease, and this is what we did. So we took a skin biopsy from a non-wounded portion of the patient. We used a retrovirus, a virus that can take the 9 kb collagen 7 gene. The virus infects the patient's skin cells, and now the skin cells produce collagen 7. So it's the patient's skin cells, but now it contains the wild type protein. It takes 25 days to grow up the patient's gene-corrected skin cells, and it looks very, very thin. That's what the epithelial layer looks like. And from 2 punch biopsies, we can make 6 of these gene therapy grafts. And these gene therapy grafts are supported just on Vaseline gauze, as shown here. Then we take the gene therapy grafts and take the patient to the OR and sew on their new genetically corrected skin. That diagram was really easy and straightforward to explain. The bad news is it took us about 20 years. And maybe if I had some AI agents as a professor and a lab and a technician, this could have taken 5 years. But back then we didn't have AI agents, and this was reality. We were funded by NIH and the patient foundations. And you read about these technologies and you think about the end and the patients who are treated. But what's amazing about this story is it came out of the Stanford labs just across the street. So they created the mouse model for EB. They took the mouse skin, put in the gene, made sure it healed the wounds. But Stanford is not a pharmaceutical company or a biotech company, right? So in order to put the gene therapy grafts into patients, you can't just grow them up in the lab. We had to create our own little bio bubble and a small GMP facility just to cleanly and accurately grow these skin grafts. So with all of the testing, we submitted an application to do the clinical trial. We treated the first patient in 2013. That was only 4 patients, and I'll show you some of the data. And then when we got positive results in the phase 1-2, then we could attract a partnership with a commercial partner called Abeona. Abeona then took over the IP and then paid for the phase 3 trial. And the good news is in 2025, last year, the drug was approved. So now it's commercially available to these rare patients. This is the phase 1 result. So this is a young man with huge wounds on his arm, and he received the gene therapy skin grafts, and you see the little dots of tattoos because it marks the corners of where we put the gene therapy skin grafts. So you see the open wound, and 6 months later you see a healed wound. Certainly the skin is still pink, it doesn't look normal, but at least the wounds are closed, not painful, and not itchy. So on the basis of that, we did more patients, and as a phase 1 trial, the first important endpoint is safety. We wanted to make sure the virus didn't spread out of those gene therapy grafts and infect the brain or liver. It didn't do that. We wanted to, these patients don't have the normal collagen 7. So what if they developed an antibody response and had an immune reaction against the new protein? We didn't see that. And so these individuals have multiple wounds. It's a rare disease. So even in the phase 1-2 trial, we only had to recruit 7 patients with multiple wounds. And because they have multiple wounds, we had assessments on 42 wounds total. And so in the bottom panel, you can see a large wound on a young woman's arm. After surgery, 2 of these gene therapy skin grafts were placed. 9 months later, you see healing of the wound. And the molecular markers where we did serial biopsies at the beginning, at 3 months, 6 months, and 12 months, the red arrow shows now the restoration of the collagen 7 gene. So what this means is we see skin healing, and that skin healing is due to the restored collagen 7. So it's really nice to grow the grafts at Stanford, to have a small GMP facility, but now when we're doing the tech transfer out to a different company, things are very, very different. So the GMP facility was on California Avenue, and one of the biggest challenges was taking the patient's gene therapy cells and transporting them to the hospital, and the gene therapy sheets are in cell culture media. You know, we drove the sheets. There were speed bumps on California Avenue, so you have to make sure the media doesn't splash. You can't imagine all the technical challenges and hurdles to actually manufacture a product. Now we transfer the technology to the company Abiona, and now the manufacturing plant, instead of 200 square feet, it's 10,000. It's in Cleveland. And when the patient comes and sees a dermatologist, we biopsy the skin, send it to Cleveland, and in 25 days later, a technician comes back from Cleveland with 6 to 12 of the patient's gene therapy grafts. So this was a project and a passion project for many years with a small group of scientists. One incredible challenge that I never realized being in academia is when you transfer the asset or the drug product to a company, you are subject to the whims of Wall Street. Okay, so when Wall Street biotech does well, you do well. When biotech doesn't do well, you crash as well. And so we transferred the technology and out-licensed it in 2016. Over that time, 4 CEOs passed, and that was very painful because they had to raise money, we had to get introduced, we had to work with a new team. But I wanted to acknowledge the 2 scientists that stayed with the program for over 20 years. And these were the individuals who were so committed to the patients and the drug. So a phase 3 trial is the last part where we demonstrate hopefully efficacy over the control. And in the phase 3, all we had to show was that the gene therapy grafts led to greater than 50% wound healing compared to baseline, and that it reduced the pain on the wounds. There were 2 sites, Stanford, University of Massachusetts, and when we enrolled 11 patients, because these 11 patients have multiple wounds, we had 86 wounds to assess. So here is the technician who flies and brings the gene therapy skin grafts. It's a cardboard box that they hand carry to Stanford. The patient shows up at Lucile Packard Hospital, where the plastic surgeons debride the wounds and clean it up, and the wounds have to fit the 5x7 rectangular shape of the gene therapy grafts, and the surgeon applies the wounds, and the wounds stick with the absorbable sutures. So in this particular patient, she received 6 of these gene therapy skin grafts. These are the published phase 3 results. The average patient's age was 21. The take-home message was that 43 wounds were randomized to the gene therapy graft. 43 matching wounds were randomized to control, so no treatment at all. We followed the wounds over time to see whether the gene therapy grafts would heal wounds compared to the control. And I apologize, this is probably some of the worst images, but this is a large chronic wound that's been present 10 years on the thigh of a young woman. On the day of surgery, the huge wound is debrided and cleaned, and 3 gene therapy grafts are placed. At week 24, you see healing and closure of the wounds, and the little black dots represent the tattoo marks that show the edges of the gene therapy graft. And so what's incredible is this individual went from 10 out of 10 pain on morphine to 0 out of 10 pain when that wound was closed. And that result has been persistent for the past 4 years. With any gene therapy trial, we have to commit to the FDA that we follow these patients for up to 15 years for safety. The lower panel shows smaller wounds that also received the gene therapy graft, and you also see healing here. This is just a summary of the results. The red bar shows the gene therapy skin grafts, the gray bar, the controls, and the graph on the left-hand side, it shows the proportion of wounds at week 6, week 12, week 24 that reach healing. And you can see the red bar shows that the gene therapy grafts healed the wounds much better than the control wounds. The right-hand side graph shows the decrease in pain at week 6, at week 12, and week 24, the gene therapy graft reduced pain by 3 points and more than the control. If you had an endpoint of greater than 75% wound healing, the gene therapy skin grafts also outperformed the control. This last column here shows the reduction in itch as well from the gene therapy skin graft compared to the control wounds. So that took 25 years, and there aren't many patients. So the challenging part is now it's not that Stanford and other centers are qualified treatment centers. So there's probably 6 qualified treatment centers where patients have to fly to these treatment centers, get biopsies, and do the surgery. It's cell-based, so it's time-intensive, labor-intensive, and like many of these gene therapy drugs, cost millions of dollars, unfortunately. So far, Medicaid, Medicare is willing to pay for these costs. The big question in the gene therapy field is, as more and more drugs get developed, how is America, how is any country going to be able to afford that kind of price tag for cell therapy? So the next frontier is really to use a different approach. We're going to use CRISPR gene editing, where now it's a lot cheaper to create nucleic acids. Hopefully we can spray on the gene therapy CRISPR enzymes into the wounds and correct the cells and heal the wounds. And we think that hopefully will be a better price tag and a cheaper drug. These patients I mentioned have more than just skin problems. They have eye problems, scarring, GI problems. And now we also have a research project where we use and biopsy their cells, use the Yamanaki factors. We can change them into their stem cells, induced pluripotent stem cells, use CRISPR to correct those cells, and then use different agents and factors to differentiate those iPS cells into epithelial cells of the eye, epithelial cells of the gut, and hopefully treat the multiple organ systems that are damaged in the disease. I wanted to acknowledge the scientists, the collaborators. This research was actually brought by Eugene Bauer. So maybe some of you were medical students under Dean Bauer. This is a patient that we treated with the gene therapy skin grafts, and for the first time ever, she was able to go to prom as a high school student. So this is a rare disease, but I think it's important because it demonstrates the power of genetic therapy and gene editing, and I think you're going to see more and more of these examples in the future. It starts with rare disease, but one day, hopefully, if there's important genetic causes for things like Alzheimer's or Parkinson's, that those diseases may be amenable to gene editing and correction. I'll close by saying that, maybe some of the younger medical school alumni know Eddie Vedder and Pearl Jam. He is an incredible benefactor and supporter of EB research. And so if you want to learn more about our research, about the disease EB and the Patient Foundation, there was a documentary produced called Matter of Time. It won awards at Tribeca, and now it is on Netflix. So it's called Matter of Time. And if you're interested in learning more, you can watch this movie. And so that's it. Thank you very much. Open it up for any questions or anything.
I was actually going to start off with a question. What happens to these patients if there's not effective treatment? What is their life course?
Right. It's of incredible suffering. The wounds are large. They're painful. When the parent goes and washes and changes the wounds, the dressing itself takes about 2 to 3 hours. So it's the parent almost torturing the child, and the child doesn't understand how important it is. You got to heal the wounds. Over time, the child gets enough wounds that, it's raw, it's painful, and the wounds cause anemia. So some of them die of cardiomyopathy. Some of them die of systemic infections. If they survive past their 20s, because the wound has had so much inflammation, many of them pass from squamous cell carcinoma that grows in the wounds, and those squamous cell carcinomas are very aggressive. They metastasize. They usually pass in their 20s and 30s.
Yes. It looked like in your therapeutic pictures that between the grafts there's some healing. So it's
Yes.
migration of the cells.
Yeah,
Maybe a strategy for sort of
yeah.
seeding or something.
Yeah, we hope we don't know if there's migration because, with a sample size of 11, we weren't able to biopsy all the periphery. But what we think is happening is you're basically creating from a large wound a smaller wound. And so maybe the edges, because the central areas have been corrected or healed, now the smaller edges, it's just easier for epithelial cells to migrate and close. When people ask, well, if there is wound healing for years, the skin sloughs off in a month, right? So how can you have good effects like this? That means the virus must have transduced and corrected some of their basal stem cells, right? So another approach is really to collect more of the basal stem cells, and hopefully there's more durability. So far, commercially, about 4 patients have been treated so far, and we'll see how this does, and how this scales. We're always scared because of Wall Street. We're always scared, if a lot of these gene therapy companies will survive, because it's a very difficult market. If we were doing gene therapy like GLP-1s, that'd be different, but this is a different game. Yes.
So,
Hold on a second, I can give you
Oh,
the mic.
sorry.
I mean, how do you plan to address the systemic problem of dermal and especially inaccessible areas like the GI tract?
Yeah, absolutely, absolutely. So this is only cutaneous therapy, right? It was the first step.
I mean, the whole dermis is at risk, right?
Yeah,
So,
exactly. So the hope one day is to do CRISPR gene editing. So the CRISPR can be delivered as messenger RNA for the CRISPR-Cas enzyme. The guide RNA is specific to the patient's cell. And so our next research project, and it's encapsulated in a skin-targeting lipid nanoparticle, similar to the COVID vaccine. So the goal is to use microneedles or lasers to create little holes in the non-wounded skin, but still defective, and hopefully spray on the CRISPR gene editing tools to reach those cells. That's, again, skin only. If one day we are able to create an epithelial homing LNP, then you could do it systemically. And that's the holy grail. But so far, for lipid nanoparticles that encapsulate the CRISPR nucleic acid, right now the homing is only to hepatocytes, right? So first pass clearance in the liver. We don't know how much will get into the skin, but that is the next step. Thank you for that question. I know we have audience members from biotech and other places, so that's always a lot of fun. Question? Yes.
Do you worry about any sort of, is there transfection in this process? And do you worry about integration and possible nuclear deterioration and malignancy from the treatment?
Absolutely. That was the phase 1, and also these patients are followed on for 15 years. So far, we haven't seen it, and your idea is absolutely right. The retrovirus may insert into an oncogene, knock out a tumor suppressor, create a teratoma, and one of the biggest negotiations with the FDA is to monitor the sites, and if we see any suspicion, we take out the graft. So far, that hasn't happened. The caveat is, we've only treated 18 individuals, so they need to be followed. Now in gene therapy, people use adenoviruses, lentiviruses, maybe a little less risk with that, but still there is that risk. So, yes, please.
Hey, thank you for this. So I'm one of those biotech folks, and I have to say, watching this was inspiring.
Thanks.
Right? I mean, you spent 20 years of your life, and I can just see the fighting you've had to do over the course of that timeline to get to where you are now, and the results are amazing for patients, right? So thank you for that. I mean, and I have to say, it's funny, I'm a biotech CEO, and I'm the 3rd CEO of the gene therapy biotech, so I can also relate to what you're describing, right? It is extremely tiring. Right, to stay on track for the mission. Right. So I guess the question I had to kind of shift all the way to the other end, because I think a lot of these questions have been scientific or clinical, is at the end of the day, if we don't solve the business side problem, this isn't going to work.
Absolutely.
Right? And I have to say, as being someone who was out there fundraising last year, as soon as when I was talking to VCs, maybe some of you were in the room, right? As soon as I said the word gene and therapy, they ran for cover.
Oh, no.
Right?
Yeah.
The field itself,
It's unsustainable.
I think the phrase I
Yeah.
heard from some of the founders in some of the interviews last year was the gene therapy business model is fundamentally broken. Too expensive to develop it, too costly on a patient per patient basis.
Yeah.
At the end of it, after all that investment from a VC, you're treating 10, 20 patients a year, which
Yes.
means millions of dollars from a business standpoint per patient,
Yep.
which is not sustainable,
Yep.
right? And so I do think I do worry, especially with all the cuts, I worry about the state of research in general.
Right.
But then, in this field, how do we get to a spot where this works? Because there's so much more that has to be solved. I love the fact you're bringing up LNPs, right? That's sort of the next delivery technology.
Yeah, yeah.
But how do we make this work? If the investment doesn't continue to go into the field,
Right.
we're not going to help the patients that we want to. So
Right,
just
right.
love to get your thoughts on that.
Yeah, it's incredible questions. I approached this scientifically, saying, here's the missing gene. We need to put back the protein. We can do it. We just push, right? One story I didn't tell you is the vector, the retrovirus. It's one thing to use an academic grade from Indiana Vector Facility, but Abeona, before knowing the phase 3 results, had to make a GMP center to manufacture the vector. That's $70 million. That's a $70 million bet before you even know the outcome. So to your point, it's incredibly high risk. And then at the end, it's about market access. And right now, there's a lot of, as drugs roll out the first 2 years, a lot of hand-holding, trying to find the patient. We're going to make it easy. We're going to give you travel vouchers. We're going to make sure you get the therapy. And, for many of these companies, as you said, you need to treat a minimum of 10 to 20 per year in order to break even. If you don't hit it, the company, will dissolve unless they, attract some other product. To your question of what about the future? How can we make this so sustainable? Like many of you, I'm not excited about HHS and the leadership there, but maybe one change is that for gene therapy and gene editing in rare diseases, they are coming down with the FDA regulatory requirements. Is that enough to make it cheaper? I'm not sure. But all of the animal toxicology studies, all of the safety studies, that is predicated based on information and requirements with small molecules. That makes sense. It's cheaper, a little easier to make. But when you think of so few patients and how expensive gene therapy is, they are coming down with the regulatory requirements. So in the New York Times this past year, some of you may have read about baby KJ. This is a little baby at Children's Hospital of Philadelphia with a urea cycle disease of the liver. In 7 months, they created CRISPR gene therapy to cure the patient, LNPs homing to the hepatocytes, right? And so right now, the government and corporations such as Danaher, they're the ones who produce the LNPs and the nucleic acid. They are trying to figure out a working model for academics and biotechs, so the reagents in the beginning can be a little cheaper, so there can continue to be investments and clinical trial development. What happens afterwards? We will see. I mean, hopefully the idea is if the timeline and the cost of reagents are less expensive, maybe the final price tag of the drug is not quite as high. But we shall see. It's so bad that so many, as you know, gene therapy companies, even if the drug works, it doesn't make money. And so they give up the drug, and the Patient Foundation as a nonprofit takes back the drug, and the poor nonprofit company is trying to somehow figure out a way to give the drug to patients. So this is a very volatile, changing, unknown landscape. And as physicians and scientists, it's heartbreaking and unexpected in the sense that it works. We fixed the gene, the patients are better. Why can't this happen? And there's so many more complicated factors. I'm curious, do you have any opinion in terms of what would happen next? Or, I mean, like, you know,
fair question.
It's the people in the room. I don't know. Well, what is your take?
I have lots of takes. I'd say first is, you know, I mean, I've got my Stanford MD and I got my MBA in large part because I did think that at the end of the day, to provide the best care for patients, we have to find a way to interact with the business world.
Absolutely.
Right. And I think this is, to my mind, probably one of the biggest examples of where that collision is happening.
Yes.
I mean, the phrase I give to investors is, you know, back when I was in med school, right? So, you know, in those classrooms, right? You know, gene therapy was a science fiction.
Right.
Right? I mean, the concept of, you know, well, back when I was in med school, we hadn't even sequenced the whole human genome, right? And this idea that we could actually identify specific genetic diseases and treat them was science fiction. And here we are. It's science fact. It's clinical fact. It's amazing, right?
Right.
This final barrier is huge. And as a physician, I think it's, in many ways, that's our obligation to figure out how to make it work.
Right.
Right? I do think the thing you're bringing about reagents is key. Mm-hmm. I also think the potential of genetic medicines to treat not just rare genetic diseases, but actually, because if you can insert a whole gene, you can actually insert genes that encode for things that are not part of the human genome, like biologics, as an example, right? And if you do that, all of a sudden, now you can actually start to treat non-genetic diseases, which creates volume at scale, which changes the business model dramatically. Right? Because part of the reason the reagents are expensive is because they're just generating enough in teeny-weeny plants that cost $70 million to build and only 10 patients a year are being treated. All that $70 million has to be placed upon 10 patients per year, and that's where the costs explode, right? And so I actually think we have to broaden our aperture in terms of what does gene therapy and genetic medicines really mean. That will require, especially for bigger populations, the FDA to think about this differently, which, to your point, they are. But yeah, I also feel there's some urgency to this. I do worry the field's gotten to a point now where if we don't solve this, I was just looking up, I forget your company's name, but I mean, their market cap for a company in phase 3 trials is really, really low.
Yes, it's really bad.
Especially given the quality of phase 1 and phase 2 data. This is the challenge that we face now. And I worry if we don't somehow fix this in the next 4 or 5 years, we're going to start to see the progress. I mean, the progress is already stalling, as you know, but I think it's going to stall even more. So I feel there's some urgency for us as clinicians, as scientists, as business people to figure out how to make this model work. Because patients, this works. Right? And so we have to figure out a way to make it work for patients, because it would be a shame if this doesn't reach its maximal potential, I think, and over the course of the next decade.
Dr. Tang, how many people in the country have this problem?
Probably in the U.S. for this type of, it's around 2,000 to 3,000. Worldwide, probably 5,000. So it's not a lot. It really isn't. And so it's a fundamental challenge. To end on a high note, the good news is, you went to a school where the infrastructure and the dean and the community invested in this. How many schools can you say, developed a gene therapy drug? So this wasn't possible, without the scientists, the infrastructures, the dean willing to build a GMP little facility. So, when people say, is translation, is genetic medicine possible? You went to a school that did it. So.
All right. Well, thank you so much.
Thank you. Thank you.